This is Dr. Phoebe and Stapleton, and also it's a good pleasure on behalf of the MOT rigor interesting group to invite Dr. Stapleton, and I actually this is the first time to meet her last dinner, but look like we have a lot of the common and interest, and this morning I talked with her, and we have a excellent common words communication. So Dr. Stapleton is currently a senior professor, is tenure track, and in the Rogers University, and affiliated to the Department of Pharmacology and the Toxicology Department, and also is the Environmental Occupational Health Center Science Institute. And Dr. Stapleton received her a Ph.D. and a postdoc training in the West Virginia and the university, and focused on the exercise physiology and inhalation toxicology, and her lab and focus on the implication, cardiovascular implication of maternal exposure to the nano material during pregnancy, and try to identify some maternal or fetal and offspring and outcome associated with maternal exposure. And she's awarded and in 2021 for the outstanding new environmental science and called the ONCE grant. Congratulations, that's great. And she also got the university award, council award, and that is a counselor basic science research, and the short name is BRBHS. So that is excellent. And her most work and focused on the inhalation of titanium and one metal and dioxide and nanoparticle, but recent years and her lab and start to interesting and toxicology outcome and associated with the micro and nanoplastic particle and exposures. And I think she have very good publication, more than 35, and so congratulations. And so welcome. Yeah.
Thank you. Thank you so much. Thank you very much for the introduction and very much for the invitation, Dr. Stakes, Dr. LeBlanc. So thank you very much for having me here. I was able to come just before COVID, so I've been able to meet with a couple of people that I saw when I was here before, but we've also changed our research. Not a lot, we're in the same model, but we're using different materials now and different considerations based on those outcomes. So last time I did a lot of talking about titanium dioxide, and this time we're going to transition to micro nanoplastics. So a little bit of a review just to make sure we're all on the same page as we continue the talk, but then moving into some of our new stuff. So with that, our lab started with the question how something one inhales can affect the cardiovascular system. So you may be familiar with this idea that air pollution and particulate matter through epidemiological studies have indicated that there's epidemiological evidence of cardiovascular effects of particulate air pollution. And that's where the morbidity and mortality rates are coming from these high air pollution exposures. So increased incidence of myocardial infarction, angina patients, heart-related issues within 24 hours of inhaling particle pollution, or within 24 hours after a high smog day in the city, we'll see this increase in ER visits, which then increase morbidity and mortality. Interestingly, there's also an increased risk of intrauterine growth restriction or fetal growth restriction in women that live in high pollution areas during their pregnancy as well. So this idea that exposure to air pollution has cardiovascular effects, and those cardiovascular effects may also play a role in reproductive health. But overall, the concept that exposure to fine and ultrafine particulate air pollution has adverse effects on cardiopulmonary health. When we talk about particulate matter, we need to make sure that we're talking about the same size range. So particulate matter has 3 fractions. There's the coarse PM or PM10, which is indicated here at the top. And so these are going to be the larger things that you can see. So this is the dust, the pollen, mold, things like that, less than 10 microns in diameter. Then there's a fine PM, and that's going to be PM 2.5. That's the regulatory standard for EPA measurements. So when you talk about a high particulate matter day, it's the PM 2.5 that they're actually discussing. And that's going to be less than 2.5 microns. And in this case, you can't see them as well, but they're what make that black plume of smoke behind a diesel truck, for example. So combustion particles, wood smoke, organic compounds, metals, things of that nature are going to be in this fine PM 2.5. And so what you might notice is you can probably see these big black dots as you look at the screen for the coarse. You can kind of make out a gray haze for the fine there, but there are dots there. And then there's this idea of ultrafine particulate matter, which is PM 0.1. So less than 0.1 microns in diameter, which is also identified as 100 nanometers in diameter. So when we talk about nanomaterials and ultrafines, we're talking about a particle that's within the same size range. So these are invisible to the naked eye. So there really are dots here. You can come check the screen later if you want. But the idea is that these are things that you can definitely see. This is a gray haze, and these ultrafine particles are invisible despite the fact that they're there. So again, this is within that nano range. So in this case, especially after COVID, we're talking about viruses. We're talking about very, very small end of the bacteria. We're talking about antibodies, definitely things that we can't see, but that's the size range that we're referring to. These ultrafine and nano-sized materials and particles come in a variety of different shapes, sizes, and chemical compositions. So we can have diesel exhaust particles, which would be incidental, and we can also have highly engineered nanoparticles like quantum dots. They may be a carbon that may be in the shape of like a soccer ball for a fullerene ball shape. They may also be a carbon that's made into a nanotube form, either a multi-walled or a single-walled carbon nanotube. They again can come from combustion, so wood smoke, but they can also be intentionally applied, like the titanium or zinc dioxide that's in mineral sunscreens. They're used in a variety of different computer products, and then we can also have these materials due to just normal use. They're in the laser printers, they're in the dust that we take out of our dryer lint, and also in 3D printer emissions as well. So they're kind of everywhere we look without realizing that we're seeing them at all. And the concern about that is the toxicity associated within that size range. And we'll come back to this size range concern at the end of the talk as well. But it's known that these larger particles can come into the pulmonary space. They get trapped in the mucosal oscillatory escalator in the trachea, in the bronchi, in the large components of our lung, and they're cleared. And therefore, the toxicity is reduced because these materials leave the pulmonary system. However, the smaller ultrafine or nano-sized particles bypass those clearance mechanisms. And therefore, there's an increase in particle retention in the lung. That can lead to inflammation that can then trigger a systemic inflammation or a pulmonary spillover. There's sympathetic neural activation. And then there's a concern of these particles translocating from that pulmonary space. We think of the lungs as being a physiological barrier, just like the blood-brain barrier, the placenta, the blood-testes barrier. But in this case, we do have evidence and reasons to think that these smaller particles may translocate from the lungs to systemic organs. Now, the original goal of our work was to understand how it is something that you inhale in a particle form can affect cardiovascular function. And again, the three theories of that are going to be pulmonary inflammation, because that pulmonary inflammation may lead to a greater systemic spillover, and inflammation can wreak havoc on the endothelial layer of the vascular system. Maybe sympathetic activation as well. Those sympathetic nerves allow for vasoconstriction or vasorelaxation. And then the particle translocation will also allow direct particle endothelial interactions. We talk about the vascular endothelial space because it seems every time I give a talk or meet with an individual, we all have our organ system of interest, or at least we started there, and mine happens to be the endothelial layer. I think this is just a super cool organ that's highly overlooked. So this is a single cell layer that lines all of the vasculature, so all of the blood vessels that you have in your body, and these cells act as an autocrine, paracrine, and endocrine organ, which means they're able to talk to themselves, they're able to talk to their neighbors, and they're able to send signaling mechanisms upstream or downstream from their location. And most often, the communication is paracrine in that they're talking to the vascular smooth muscle that are the neighbors that are located next door. Now, these vascular smooth muscles either can constrict or dilate based on those different signaling mechanisms. If they constrict, the lumen of the blood vessel will get smaller, and if they dilate, then the lumen will get larger, affecting blood flow to the downstream tissues after that blood vessel. And there are a number of different chemical agonists and mechanotransduction signaling molecules that can be attributed to endothelial function or dysfunction. So when we see this endothelial dysfunction, normal endothelium-dependent dilation allows for increase in blood flow, a larger lumen, a larger blood vessel. However, if the endothelial layer is dysfunctional, that can be due to increased constriction factors or no change, that those dilation factors are not coming across, the signal is lost somehow. Then we get a reduction in that large dilation, therefore a reduction in blood flow downstream, and this is termed endothelial dysfunction, when those cell mechanisms and cell signaling factors aren't being understood or aren't being produced to begin with. So we had done a lot of work in occupational models, young, healthy male animals, and we found in these young, healthy male animals that we were getting this vascular dysfunction. And that's great from a scientific context. But I was living in West Virginia at the time, and as I looked around our state or my lab or anywhere, the number of young, healthy 20-year-old men in that area is pretty well slim to none. So we started to look around a little bit and come up with other models. So at first we used an obesity model, thinking that that would be of interest as well. And we put Zucker obese animals into our inhalation facility, just in that laboratory, what if, let's see what happens, how much exposure would it take for them to have some type of coronary episode? Well, those are so sick to begin with, nothing would happen to them. We could put them in for a long period of time and they were still sick when they came back out, but the compensatory mechanisms had triggered on. So we started thinking about what else we could put into our inhalation facility. And we came up with the maternal-fetal model because it's a complex and widely understudied model. The nuanced changes that need to happen within the female physiological system need to be rapid and precise to allow for tissue development. You have to build an entire new tissue during pregnancy, known as the placenta, in addition to fetal growth and development. There's a wide hormonal variation that can take place, but those hormonal variations are actually limited in pregnancy in compared to female hormonal variation during their reproductive cycles. And then there were these physiological disparities from normal in pregnancy. There's an increased blood volume. There's an increased heart size. The blood volume increases by about 40%. There's also an increase in tidal volume. So during pregnancy, a woman doesn't have more respirations, but they have deeper respirations. So every breath they're taking is taking in more air. And so we wanted to understand how something a mother inhales during pregnancy could affect her health, the fetal health, and that of the surviving offspring. So that was our next research question. So our laboratory model and experimental design looks something like this. We expose our Sprague Dawley rats from gestational day 4 to gestational day 19. The implantation in a rat takes place at around gestational day 6, so we're getting them just prior to the implantation stage. From this exposure, we can then look at our maternal outcomes, our fetal outcomes, and our offspring health. We use a whole body inhalation exposure system, and that is based on sound waves that are coming. So if you've ever sat next to a car that has the bass booming at a stoplight or a stop sign, and you can feel your whole car vibrating every time the bass goes in their car, we're using that, I'll call it technology, to aerosolize our particles here. So we actually have a speaker and a subwoofer that allow for that boom, I guess, to aerosolize our particles. And then using a variety of different airflows over the surface, we're able to pick up the smallest of the particles over the top. Everything is measured in real time, temperature, humidity, concentration of our particles, and that allows for day-to-day consistency of our exposures, but also minute-to-minute consistency of our exposures as well. We monitor the concentration, and since we do nano-sized work, we have to prove that we're actually looking at particles in the nano range because, like we mentioned before, you can't see them. So we have some tools as well to be able to monitor the size of our particles, again, for that day-to-day consistency. So from this, we've learned a lot. We learned a lot about maternal outcomes, fetal outcomes, and offspring outcomes. The exposure to these nanomaterials, the titanium dioxide during pregnancy, can lead to differential vascular responses and differential time course responses. So at different gestational ages, that will impact the uterine vasculature of the female mother, depending again on when we started the exposure, so early, mid, or late gestation. It kind of culminates to this inability to meet perfusion demands, develops into an IUGR or FGR phenotype, and that may also lead to impairments in conception or postpartum coronary recovery. And we'll come back to that component at the end. In our fetus, they have a IUGR phenotype, so they're smaller than what their gestational age should be, or fetal growth restriction. And when we look at the fetus, they have impaired fetal cardiovascular health. We've seen vascular dysfunction using the tail artery. We see cardiac and coronary dysfunction, reduced coronary bioenergetics, and epigenetic alterations as well in our fetus. And when we allow those fetus to deliver normally and look at the surviving offspring, we have outcomes from that FGR phenotype. We are able to find some of the nanomaterials in the fetal compartment. We'll come back to that. But we also see impaired adult cardiovascular health. So vascular dysfunction both in the heart and in the uterus of the female offspring. We see behavioral consequences in the male offspring. And that's not to say that we wouldn't see vascular consequences in the male offspring or behavioral consequences in the female offspring. It's just that's how we allocated those animals. So it's not that there's not a sex differential. We just haven't looked yet. And then we see impaired adult cardiovascular health. Again, vascular dysfunction, the behavioral consequences, cardiac and coronary dysfunction in our adult animals, and mitochondrial dysfunction leading to reduced coronary bioenergetics. So we've done a lot of work in this maternal-fetal field. But there's this big black box in the middle of that. I guess it's really a big red box in the middle of that, which is the placenta that's connecting these two systems to each other. And not a lot of work has been done in what happens to the placenta itself or within the placenta. So our next question after finding these outcomes was to identify if these outcomes are associated with particle translocation. Is it because the particles are getting to the area, or is it some kind of change in the maternal physiology that's leading to these fetal outcomes? And so we needed to identify that before we could continue looking at our fetal offspring, our fetal and our progeny. So the question was, does the placenta act as a biological barrier, or can engineered nanomaterials or ultrafine particles translocate across the placenta? And so with this question, we developed a placental perfusion technique. We would excise out a section of the uterine horn. We would remove the fetal tissue, pull the uterine horn aside, and we could cannulate the uterine artery both at the proximal and the distal end. And we could also cannulate the placental umbilical cord both through the umbilical artery and through the umbilical vein. When doing this, we could then collect the effluent that came off of the distal end of the uterine artery, but also collect the effluent that comes from the umbilical vein. Primarily, we were looking for these nanomaterials. That was the goal. So this is proof of principle in that we put Evans blue dye into our placental system. You can see the Evans blue dye coming through. It dyes all of the protein because Evans blue dye is an albumin-bound chemical. And so you don't see any blue coming down here through that umbilical vein. And so this is the picture when we got the system all up and running. So this is my evidence of how to quickly scare a graduate student away. That I got my system all up and running. It had taken me about 2 years from daydreaming to this drop coming off of the umbilical vein, which meant the whole system was working. And we had cross perfusion coming through the placenta, at which case I might have had an outburst that did not use profanity, but it was boisterous. And that was the time that my rotation student decided this was not the space for her. So she has since graduated from another lab and is doing very, very well, but it was not in my space. So, but the other graduate student thought this was a super cool idea and she took it on. So she recently graduated in January as well. So from this, we added a bolus dose of polystyrene nanoparticles, 20 nanometers in size. They were rhodamine labeled. So we added this bolus dose into the proximal uterine artery, and then we just collected that effluent over the course of the next 3 hours. And so we used a plate reader with that rhodamine tag. We were able to use a plate reader to be able to identify that increase in fluorescence, which would identify increase in polystyrene concentration coming through. So before anyone asks, we did do the experiment that we took the effluent and we centrifuged it down and then took the supernatant, and there was no rhodamine in the supernatant. So we know that the rhodamine was still attached to that polystyrene bead as we're going through. So this is representative of those beads. And what we see is a significant increase in the uterine artery about 10 minutes after this bolus dose goes through. So it passes through the uterine artery very rapidly, and that concentration stays high to about 90 minutes. So about an hour and a half after adding that directly into this uterine artery, it's all passed over the placenta. When looking at the umbilical vein, we see not much change happening over the course of those first 90 minutes. And then we see significant increase in our rhodamine label through the plate reader, our fluorescent intensity after 90 minutes. We start to see these peaks coming through. And not only that, I think it's just off the screen, but we use a 3-inch stainless steel blunt dissecting needle to cannulate the umbilical vein. So we removed that and ejected all of the fluid that was still in that needle because it has to drop out like I showed before. So when we take all of that out, there's this remnants down here. It's just off the screen. So we are able to identify the nanomaterials passaging across the placenta in this very controlled environment. So I presented this to our center well before COVID at this point, because I was really excited about getting this placental perfusion technique set up. And a colleague came to me at the elevators as I was leaving and said, I really need you to come talk at this micro nanoplastic session that I'm going to be giving in 2 months. And I said, I don't understand why I'm going to be doing that. I do titanium dioxide nanomaterial exposure. And his comment was, you know, you're doing plastics, right? Polystyrene is a plastic, and therefore you're doing plastic research. And I said, no, I just want to know that my particle size is getting across. And he said, well, that's lovely, but could you come and give a talk about what you just did at our meeting as well? And so I did. And that opened a different Pandora's box that I never knew I was looking for. So not only does the placenta not act as a barrier to nano-sized particles, but it also does not act as a barrier specifically to nanoplastic particles. It also doesn't act as a barrier to titanium particles, nor does it act as a barrier to gold nanoparticles either. So 2 goals with 1 technique. So after that conversation outside the elevator, you open your eyes a little bit more and you start to realize that we are just surrounded by plastic. Even looking at the room, the covering over the tables to make sure that they're easier to clean is made out of a plastic. The carpeting is made out of a plastic. The seats are made out of a plastic. The shields to keep the sun out are made out of a plastic. And I haven't started with the things that we have in front of us yet. So that was just walking into the room. So you start to realize that it's everywhere at this point. And the lifetime of plastic is only increasing in exponential forms, exponential timelines, I should say. So from the 1950s, now this is only out to 2015, but you can appreciate that we're only finding more ways to use this product and finding fewer ways to eliminate this product. So the breakdown of plastics ranges from about 10 to 1,000 years. This is the estimated breakdown. And that breakdown consists of thermal, UV, and mechanical degradation. So in order to make a polymer, you have to take a single monomer, change its chemical structure slightly to be able to add them together. So when we talk about this breakdown, we're not really talking about decomposition from a polymer back down to a monomer. We're just talking about this particle getting smaller and smaller and smaller until we can't see it anymore, and then we call it gone. So this is the concern, because an average estimate for a plastic bag degradation is about 20 years. So many of us have been around to be able to, plastic bags have broken down during our lifespan. Maybe that's the best way to say it. But a plastic bottle to break down is estimated to be about 450 years, which none of us will be around to see this plastic bottle breaking down. So again, this concept of decomposition is unclear because we're not referring to this chemical degradation, but often are referring to particle size instead. And when we look at microplastics in the environment, many of the air pollution studies have been focused on metals. So conveniently, we have, and this was a study that was done out of the lab I did my postdoc work in, we have a clean filter, we have a filter after a mountaintop mining site exposure. We take this filter, we take all of the black off of it, we run it through ICP-MS, and magically, all of this adds up to 100% at the end. And there's no other category that's in here as well. So we were able to take those monomers, we were cutting them down, and somewhere some of those plastic particles are in this, or they just weren't assessed because you can't assess it with ICP-MS technologies. Looking at national parks or wilderness, especially out west, they've started to collect the plastics that are there, and you can see just the massive amounts of plastics. This is meters squared per day. Visual counts of metric tons of plastics per year, and metric tons based on FTIR analysis. So using Raman technologies to be able to identify just how many micro nanoplastics are in these natural habitats. They're also in our homes, similar to what I just identified here. They're in the furniture, carpeting, clothing. They're for the dryer lint that's coming out of our dryer. They're in coatings, both on the external and the internal. So the internal part of metallic cans was a big deal when we had conversations about BPA and how BPA was lining our cans. Well, the plastic is still lining the cans, but the BPA has been taken out and transitioned to something else. And then lastly, personal care products as well. Then in the things that we might be consuming, so bottled water, beer, wine, tea, specifically using tea bags, rice, sugar, and honey. So even the things that we consider to be more natural, like honey, we're able to find micro nanoplastics in. And this is especially concerning as humans are spending more and more of their time indoors. So in 1979, it was estimated that people were spending 70 to 90% of their time indoors. That's before COVID. So you can imagine how much time people are now spending indoors. So to talk about micro nanoplastics, we have to have a definition of this as well. So micro nanoplastics are not a specific type of plastic. There are many different types of plastic, but it's a piece of fragment that's less than 5 millimeters in a single dimension. So this might be the length or the size of a sesame seed, for example, but the toothbrush that you used this morning to brush your teeth had plastic bristles on it. And those plastic bristles are also microplastics because of the width of those plastic fibers. Nanoplastics are a little more difficult to, they're a little more difficult overall, but in their definition, they are less than 1,000 nanometers in environmental studies, and that really aggravates all of the nanoscientists because in our laboratory studies, to be identified as a nano-sized particle, it needed to be less than 100 nanometers. So there's still this discrepancy between environmental scientists and laboratory scientists as we're looking at nanoplastics. And the concern is the increase in surface area in the nano size compared to the micro size. Okay. So our new hypothesis was that nanoplastic particles can migrate from the mother to the fetal tissues after exposure. So we had done that with the placenta itself, but now we needed to do it in the whole animal and see if it actually got into the fetal tissues. And we did that in 2 different ways. We used nano-sized polystyrene in both studies, and we either intratracheally instilled the particles and 24 hours later sacrificed the dams, or we gavaged the animals. So these are our dosages down here. And what we were able to find 24 hours later in our fetal samples, so these are not the mom's tissues, these are the fetal tissues, we were able to find particles in the fetal liver, lung, kidney, heart, brain. And then after gavage, we were able to find them in the placenta, the fetal heart, the fetal kidney, and the fetal liver. So what we're looking for are those stars, the bright stars that you can see, and we identified all of these using Cytoviva dark field microscopy technologies. The theory, when we see these jumbled up together like so, is that they may have been taken up by a macrophage, and the macrophage is collecting them. So that's what we think this grouping of particles may be due to. But we can still see the single particles throughout as well. So in all of these different tissues. So that was also helpful, but I think I've had a couple conversations that reviewers really are looking for low-hanging fruit at this point, at least in some of the reviews that I've seen. And so intratracheal installation is lovely, but no one is forced in real life for the intratracheal instillation. And the same thing for gavage. We might be consuming them, but gavage is forcing that in. So could we have a more natural way to evaluate this micronanoplastic toxicity? And so we went back to our original inhalation model. And so based on epidemiological studies, identifying the development of FGR after exposure and particulate pollution, specifically the smallest size fraction, we went back to our 10 milligram per meter cubed concentration, which is also on the SDS sheet for the particles that we are using. So we ordered bulk material, we put it in our inhalation facility, and this is what that speaker looks like as it starts to vibrate. So now we've put bulk plastic polyamide particles into our exposure facility. So these are a polyamide 12 particle, that's a nylon, it's a bulk powder, and we provide it at an occupationally derived dose. We started with 10 milligrams per meter cubed. This is what over the course of our experiments, it came out to be, because we originally needed to characterize our model for these exposures. And we did that using virgin female animals, all identified to be in their estrus stage of their estrus cycle, that are at 10 to 12 weeks of age. I didn't want to put pregnant animals in right away. I wanted to make sure that this model was functional before going into our pregnancy. So we can see that we do have nano and micro and nano sized plastic particles. They are approximately 3 microns in size, which is well within that micro size range, and the majority of them are in that PM2.5 range. When we look at these animals, there's no difference in the age. There's a slight difference in the body weight, but I'm not sure how physiologically relevant those 20 grams are at this point. This is 24 hours later, because these animals are also a little bit older. We have no change in heart rate, but we do have an increase in mean arterial pressure 24 hours after the single exposure to micronanoplastic aerosols. We looked at the pulmonary inflammation based on bronchial alveolar lavage, and we see no drastic increase in pulmonary inflammation. We see a significance, but again, the physiological relevance associated with that significance is mild at best. We're not seeing a smoking gun here. So when we look at our vascular outcomes, we looked at the uterine radial arteries here, and we see a blunting of that endothelium-dependent dilation. So we use this, we evaluate this using pressure myography, and we add increased concentrations of methacholine, which is an endothelial-dependent dilating agent. And so we see no dilation associated with that in the control animals, or in the exposed animals, sorry. But we know that they are capable of understanding what to do with nitric oxide because we add sodium nitroprusside, see no difference with the endothelial independent dilation. And we also see no difference in vascular smooth muscle function using phenylephrine. We also did some biochemical analyses looking for a systemic inflammation, but also looking at plastics as an endocrine disrupting compound. We'll come back to that idea in a moment. But we did see a significant increase in IL-6, a trend in C-reactive protein, and a trend in MCP1. The part that was most interesting to me is that we see a significant decline in 17-beta estradiol, again, 24 hours after this single exposure, with no change in progesterone. So we moved into our maternal-fetal model. And from this, we set up an experimental design looking at the animals at gestational day 20, at postnatal day 5, and postnatal day 21. And we'll come back to the heart in just a second, but what we found looking at their characteristics is that we have a significant increase in maternal heart weight. We have that is also maternal heart weight to body weight and tibial length to heart weight as well. So the hearts are indeed larger in our animals that were exposed from gestational day 4 through gestational day 19. This is not an outcome that we had seen in our titanium dioxide work. We also have a significant increase in the number of reabsorption sites. Reabsorption sites in a rat are analogous to miscarriage in a human. But the number of pups were the same overall, but the pup weight was also significantly smaller in our exposed animals. And that led us to start looking at, because of this increase in heart rate, that led us to start looking at a condition identified as post or peripartum cardiomyopathy, or PPCM. I'm still working out the terminology of this, if it should be looking at PPCM, which is widely understudied, or looking at this exposure having a blunting effect to pregnancy-induced hypertrophic recovery, which it's not a hypertrophy, it's a reducing the hypertrophy back down to normal. So the normal heart can become hypertrophic due to some type of pathological outcome like volume overload or pressure overload, but it can also become hypertrophic due to exercise and pregnancy. So we know that we're getting an increase in the heart size because of the increase in blood volume, because of the increase in tissues. And we are wondering if after exposure to our micronanoplastics, if we have a shift of this physiological hypertrophy into one of the more pathological hypertrophies. So cardiomyopathy just means bad or dysfunctional heart muscle surrounding pregnancy when we're talking about peripartum cardiomyopathy. So the PPCM is initiated during the last month of pregnancy in humans or within the first 5 postpartum months as well. So our timeline here is looking at that last month of pregnancy, also looking within the first 5 months postpartum, and then we want to look at the recovery in postnatal day 21. So I'm going to not talk about the time course component just for the sake of time, but I'll tell you about what we've found in this gestational day 20 time point. The last thing to keep in mind is the diagnostic criteria for peripartum cardiomyopathy is functionally based. So it's looking at ejection fraction below 45% or fractional shortening less than 30% or some combination of the two. So it's different than other cardiomyopathies. You don't need the fibrotic tissue in order to identify this type of cardiomyopathy. So when we take our hearts out at gestational day 20 and we do some histology on them, we don't see anything changing in H&E. We don't see any type of fibrotic markers using trichome. We do see some swelling or some edema in some of our hearts, but that's been relatively inconsistent. And so we started taking just measurements of the different morphologies, I guess, of the heart overall. So we wanted to identify the area of the left ventricle, the area of the right ventricle, the width of the heart at this longest point. And so we looked at 10 different measurements from these hearts, and we identified that number 2 was significantly different. Number 2 was that length overall. And then also number 6 was significantly different, and that was the area of the right ventricle is significantly larger in when you look at or exposed to control animals. We also looked at some ultrasound in these animals, and when we look at ultrasound at gestational day 20, we see an increase in the left ventricle endo and epicardial length, both in diastole and in systole. So the length of the left ventricle is significantly larger, and also that area of the left ventricle is significantly larger in our exposed animals. When we look at the functional outcomes in this gestational day 20 time point, we see a fractional area change. Using B-mode ultrasound, there's the left ventricle volume is increased in both, and then the ejection fraction is significantly declined in our exposed animals by about 10%. Then we also have some changes in left ventricular mass as well. We've started the mechanistic studies, and the studies that relate to postpartum cardiomyopathy, the mechanisms that are most referred to in the literature at this point have to do with prolactin changes, and specifically fragmentation of prolactin using cathepsin D enzymes. So there's a theory that there's an increase in cathepsin D activity that would cleave prolactin at an unusual site. This cleavage produces a 16 kilodalton fragment known as vasoinhibin. So these are the mechanisms that are currently under evaluation in our laboratory. So that's a lot of work in a rat. And so we need to come back out to some real world considerations, moving rat to human. And so those real world considerations, one in particular is about our placenta model and human versus rat placentation. And in that, the human, the placenta is made like a big sponge, but made up of layers within that big sponge. So you have the maternal side which is providing the blood. You have the fetal side, which is collecting the nutrients. It's not collecting the blood, but it's collecting the nutrients from that side. And you have this diffusion that has to take place between the maternal side to the fetal side through cells that are known as succinct trophoblasts. So a succinct trophoblast layer is really this placental barrier that we talk about. So in the rat, we were able to find particles that had translocated from the maternal side to the fetal side. But the rat has a succinct trophoblast layer that's approximately 2 to 3 cell layers thick, depending on your cells that you're looking at. So cytotrophoblast, but also succinct trophoblast. So 3 cell layers. The human, however, has a single succinct trophoblast cell layer. So we are seeing this translocation in our rat model, which has 3 cell layers that it has to get through. So the likelihood of this translocation occurring in the human, I would say, is pretty strong. We also need to prove that humans are actually exposed to micronanoplastics. I say we, I mean we collectively as scientists, to be able to explore this more. And so a lot of work has recently come out, particularly in the last year. So micronanoplastics have been identified in cadaver samples using Raman technologies, Raman spectrophotometry. Can never say that right. Using really fancy lasers to identify plastics in human tissues. And so they have identified polypropylene, polystyrene, and polyamide. Polyamide is important, at least for me in our work. That's nylon. That's the same material that we're using in our studies. So it's been able to be identified in cadaver. It's been able to be identified in live patient samples as well. So these individuals were going under some other type of thoracic surgery and had lung samples removed. And micronanoplastics, that's not true. Microplastics were found in these live patient samples, and the same with the cadaver samples. Microplastics have been identified in fecal samples, just 5 individuals over the course of the day, or over the course of a week. So this wasn't somebody who was intentionally trying to eat as many plastics as they possibly could. This was just normal life that they, I'm sure, had graduate students volunteer for this study. So 6 or 7 days, they kept a food journal beforehand and then started collecting these fecal samples. And again, microplastics were found in all 8 patient or participant samples. Placenta is not left out of this, and so microplastics have been identified in human placental samples as well, and these are from, again, normal pregnancies that the placenta was collected after delivery. They didn't get too much demographic information to identify where these might have come from, but this was just real-world exposure. So this was a group out of Italy. And this group has now followed up this study to look at human excreted breast milk as well, and has identified microplastics using the same techniques in human breast milk. So what we know about micronanoplastics, we know that they're able to gain access and accumulate within the primary and secondary tissues. This leads to direct cellular interactions and cellular uptake, which may lead to local effects, including inflammation, oxidative stress, altered metabolism, endocrine disruption, and gene expression changes. But the clearance or excretion of these migrated particles remains unknown. So we know that they're getting in, but we don't know where they're collecting once they get in, and we don't know how they're getting back out again. So they're not being found in the filter organs, and neither are the engineered nanomaterials. They're not being found in the filter organs like you'd think that they would be. So they're not being found in the liver, they're not being found in the kidney, they're not being found in the spleen in significant concentrations. So we don't know where they're going yet. So some more considerations. We need to understand the dosing, the laboratory dosing versus real world exposure concentrations. That is a huge push at the moment. And more specifically, the micro-sized plastics versus the nano-sized plastics. The nanoplastics are nearly impossible to quantify. We just scientifically don't have the tools to be able to measure them currently. So those material properties are a concern. Again, the size, the chemical, because polystyrene is different than polypropylene, which is different than polyamide. And that's just naming 3 different types of plastics. Polyamide 12 may be different than polyamide 6 in addition. So a lot of work still needs to be done. One of my interests is having these particles act as a toxicological vector. And that just means that the particle itself has the ability to add different chemicals to it or different chemicals on it. So this could be the endocrine disrupting chemicals might have been impregnated in the plastic, because those have all been identified, not all, many of those have been identified as endocrine disrupting compounds. So we're talking about phthalates, we're talking about the bisphenols, we're talking about the things that make different types of plastic different. So these are hardening versus foam. So in the lab, you might have a polystyrene cooler that you're using quite often, but you may also be using polystyrene bottles to be able to collect things. So that hard plastic is going to be different than the foam, and the plasticizing chemicals are added to make those differences. And so those plasticizing chemicals that have been identified as endocrine disrupting compounds. And then the components that these particles might gather just in their life after going to a landfill, those kinds of things. What toxicities, what heavy metals, what other chemicals can they add to their surface before getting into the body? The tissue bioaccumulation, biological transport mechanisms, and of course, exposure population. So are children at greater risk because they have a smaller surface area themselves, but an increased exposure just because of their height and their toys and things of that nature. So with that, I would like to thank the members of my lab. Sarah did a lot of work starting this project out. She's recently moved on in the last year and a half. She's now a scientific writer who's working as a liaison between pharmacy startups and physicians. So she's now producing all of the literature that you get when you're put on a new drug. Janine Derrica recently graduated in January. The remainder of my lab includes Chelsea Carey, Tali Seymour, Samantha Adams, and our new technician, Tanisha, is there as well. And then the molecular imaging facility, pathology services, the Cytoviva group, who are amazing, and our funding sources. So with that, thank you very much for your time and coming today, and I'm happy to take any questions. Thank you.
Some other hormone levels in the both in the mother rats and in their offspring. And I'm thinking particularly of cortisol and thyroid hormone, as well as growth hormone. And did you look at the pulsatility of growth hormone in the offspring?
So that's a great question. I understand there's a group online, so I'm just going to repeat it to make sure that we're on the same page. So the question is, within our plastic work, looking at both our virgin animals and our pregnant animals, if we had looked at any of the other hormone possibilities, specifically thyroid hormones, cortisol, and growth hormones. So in the short answer, no. So we've looked at prolactin, which also has a cycle associated with it, a circadian rhythm cycle. So that's trouble, especially during pregnancy or postnatal day, because we hadn't taken the fetus, or we hadn't taken the offspring out. And so we looked at it, but I don't trust those data at all. So we haven't looked at growth hormone. We haven't looked at cortisol. We haven't looked at the thyroid things. And I think those are all really interesting possibilities of ways that the plastics could be disrupting. And I guess that's still the unknown. So I present this work and we talk about toxicology of it, but I don't have a toxicity yet associated with that. And the plastics industry, their lobbyists point out nobody's dropped dead from plastic exposure yet, which is similar to what they said about smoking early on as well. So, there's that. But so we're still looking for some type of physiologically relevant toxicity associated with the exposures. So that's a great place to look. Thank you. Oh, go ahead.
That's our issue to exposures to nanoparticles per se versus plastics. Because I don't know, you know what I mean? Because so I wonder if you have seen the similar findings with other inner migration outside of the Pharm lab.
Yeah.
Have you considered?
So the question was about if it's really due to micro nanoplastic particles in particular, or is it due to a xenobiotic ultrafine particle, likely another metal or a carbon black, something like that. And we haven't seen that. We haven't seen those same We have seen some outcomes that are similar, and we're now starting to see some outcomes that are different. So I went back to all of our titanium work, and when we did weigh the heart, which we had no reason to, but when we did weigh the hearts for some of our pregnancies, we don't see any significant differences in the heart weight of the mothers after the exact same exposure. It's just the material changed. So it's GD4 to GD19. We're still aiming for that 10 milligram per meter cubed concentration. Same breed of rat. So it really was, we were very fortunate that it really is just the particle itself, and we don't see those heart changes in the titanium animals, only in the plastic animals. Sure, thank you. Yes.
So one of the most fascinating findings was how the heart hypertrophy happened, right, with the pregnancy and the exposure. So how we look at longer term, outcomes from that. So after postpartum, multiple pregnancies, what are the outcomes of that?
So that's a really great question. And I intentionally met with Helen earlier this morning to pick her brain because there's not a lot in the field. And we did look at some of the time course. So we do have data from postnatal day 5 and postnatal day 21. So that's approximately 5 months in a human and approximately, what is that, just under 2 years, I guess, and to extrapolate it back. And so I only learned about this multiple pregnancy question about 2 hours ago this morning. So I need to look at that. We haven't done that. We've been ordering time pregnant animals coming in. So I can't say that we have been looking at it, and I can't say that we haven't been looking at it because I only know that Charles River sent me a gestational day 3 pregnant rat. So I don't know if she had a litter before. I could probably stain the uterus and identify those kinds of things, but that hasn't been one of the questions that I've asked. So now I need to go back and find initial pregnancy and a breeder that they have been, somebody this is not their first litter. So that's a great question. I haven't done that, but we do see changes in the postnatal day 5 animals, and we do see changes in the postnatal day 21 animals. So not to the same extent as what I presented for the gestational day 20 animals, but we do see some compensation at PND5, which is something that we're continuing to explore as well. So, yeah. Yes.
For data on the sex hormones affected by microplastics, you showed that E2 certain levels of E2 decreased. Did you look to see if that's due to the fact that E2 was not being released, or is it having an effect on E2 synthesis?
I don't know. I think that's another fantastic place to look because I'm really pushing this whole endocrine disruption situation in plastics. Because we can add the chemicals, but if that plastic is getting somewhere, sorry, I didn't say anything to the people on the computer. The question was about the estrogen changes that we saw and if that was related to synthesis or if that was related to function. Is that correct?
Concentration, or is it just not being released?
Okay. So synthesis or release of it. So I don't know. I think that's a good question to ask, but it's one of the reasons that we made sure that our female virgin animals were indeed in the same stage of estrus. We're very careful about that. So they were all in late estrus, early meta-estrus when they were sacrificed because they were all put into the facility in their estrus stage. But I can't speak to why we have a lower level of estrogen in those animals. Interestingly, we don't have a lower level of estrogen in our pregnant animals at gestational day 19. So, and that 17 beta estradiol is crucial during pregnancy. So that data came in last week and I haven't had a chance to fully vet it to make sure that there's no significant difference. So it wasn't integrated into the presentation here, but I don't know. So, and I don't know what that might mean for conception as well. Yes, Helen.
Prospective mom and female throughout a lifetime will have several exposures to particles. Have you considered like doing a longer exposure upfront before pregnancy?
So we haven't yet. So that seems like a new can of worms that I would be jumping into. And we also haven't looked at the offspring yet, and the offspring health and the offspring pregnancy and the offspring sequelae that goes along with a whole new being. One, I struggle with that because I don't know how to count them. So I mean, it's really a logistical issue because do you count them based on the mom? Do you count them based on them being themselves, even though they're different groups? There's some logistical issues. But to answer your question, we have not started the preconception exposures yet. So another avenue.
Age that were out there, have you considered aging them for the faster maternal age or the risk factors?
So no, because I was kind of surprised by this outcome. So the question was if we had considered aging our animals, because advanced maternal age, in addition to advanced number of litters, are both not prerequisites, but can heighten the possibility of developing PPCM. And we haven't done that yet, mainly because I was not expecting this to be an outcome. So we never saw it in our titanium dioxide animals, and all of a sudden here it was. So that's really why I met with you earlier, because I don't know what direction to go from here. It's just so widely understudied that it was new, it was different, it was kind of cool. It's been consistent, so there's that. And then the second half of that is that we haven't looked at any of the fetus either. So we have another fetal growth restriction model. And so there are lots of questions there. So, yeah. Yes.
In one of your earlier slides, you mentioned that the offspring of exposed mothers had some behavioral changes in adulthood. Can you elaborate?
Sure. So the question was about our offspring behavioral differences. So we have two. I have one that's been published and I have one that's anecdotal. So the published one was using the Morris test. Is that correct? The Morris mazes. And then also how brave the animals were, for lack of a better term. They don't like being exposed. So the crazier they are, the more exposed they like to be. But the animals that we had, the significant difference was within working memory. And that was explained to me that they would park their car in the garage and then come to this and then not remember where they parked their car. So that was the working memory issue that these male animals had after gestational exposure to titanium dioxide, and they were approximately 8 to 12 weeks of age when those assessments were done. But again, I can't speak to the female animals because we used them for a different study. And the other was anecdotally, and that is that our female animals both started to develop some seizure activities, startle reflex. So you'd come in, we did a long-term study where we kept them for 12 months and looked at systemic inflammation, dyslipidemia. We looked for everything that we could to identify cardiovascular disease in the offspring. And we were able to identify it with endothelial dysfunction, but not anything big. And so at about 4 months of age, and I say from 4 to 9 months, because I think we sacked them at 9 months, but they would have a startle reflex. So if you went in and kind of like clapped by their cage or went to change their cage, then they would display seizure activities where they would just freeze, stop moving. So we didn't publish on that because there were 5, I think, that ended up with that seizure activity, but all of them were in the exposed group. Hi, Alex.
Great work. I was kind of wondering about the actual exposures of humans to airborne plastics, and kind of looking at the O and the EHSI up there, the occupational aspects. Is there potentially an occupational study that could be done that you're chasing after to look at like textile workers or and exposures to the plastics to kind of better estimate what is being inhaled?
Yeah, so there are those opportunities. I am personally not exploring them at the moment, although you are absolutely correct that my colleagues, I'm sure, have a connection with somebody to be able to help with that. I know that NIOSH did some of those studies in, Dale Porter did some of those studies in I want to say early 2000s, maybe late 90s, looking at, they're called flock workers. And there's a disease known as flock workers disease, where these are the individuals that apply the velvet or the velour to the surface of those types of materials. And so they had their own disease at the same time that they were doing a lot of work for asbestos and a lot of work for silica. So they did some exploration in that disease. So there are human conditions based on occupational exposures, but they only focused on the pulmonary component, not necessarily the systemic outcomes.
On that note, is there anything surfacing about mass use as a source of plastics inhalation? Should that play into these kinds of circumstances?
I think we'll probably see some of those starting to come out. But I haven't seen them yet because that was the topic of conversation during the mask use, but then also all of that disposal as well of those pieces. But no one's willing to just give us a sample of their lung that we can then use in a super fancy laser and see how many fibers you have in your lungs currently. So that's going to be a struggle to be able to identify where those plastic particles came from once those samples are available. That's a concern. Yes.
Yeah, I'll quickly comment that how basically the challenge about maybe the control panels, because these animals are born and then brought up in plastic cages all of their lives. So they're already exposed, and you're adding on top. Yeah, you are comparing to them, but what would be the true control animals that I know?
The question or the statement, I guess, a combination is about a true control for these types of exposures. So I have two statements, three. So one is that, thank you for that comment, because now I have a picture that I'm going to be adding in instead of some of this at the end for questions. Because when I was doing the ultrasound work, our animals had to be transferred down from our facility down to the molecular imaging core. And so you have to pay, not IACUC, but ACFC to transport your animals, and they can't be left without fluids. So they put those gel packs, you guys must have them here. They put those gel packs in for fluid, which is great, except it's a lot faster to just rip the foil off and put the whole thing in. So the next day when I came down to get our ultrasound animals out, and our bored rats had flipped over the gel pack and had just been chewing on the plastic the whole time, I thought, well, this is, let me explain my hypothesis one more time, because this is a concern with our exposures. And so I took a picture so that I could yell at the animal care people or explain my hypothesis, and maybe in a stronger terms. But I have that picture. So next time I'll put that in just to do this question. But secondly, it's not limited to the plastics. So we did a study looking at titanium dioxide to be able to identify how much of the titanium dioxide using ICP-MS had transferred from the mother through inhalation into the fetus and the fetal tissues. So we had all these fetal samples, we had all these tissue samples, we digested them all down, we put them in the fancy ICP-MS, and our control animals had significant amounts of titanium in all of their samples, which made it really hard to compare. And so after some of those what if and oh no and how do we deal with this conversations, we went down and took some of their food and did the same digestion and ran the same ICP-MS and found unbelievably high levels of titanium dioxide, I'm assuming, but we only have the titanium because of the ICP-MS. So titanium levels in their food, which is likely used as a filler for them. And so in great PI training mode, we quickly pivoted our hypothesis to be looking at both ingestion and inhalation exposures, because as you're pointing out, our controls were not true controls. They were getting significant amounts of titanium through their diet. Luckily, as our inhalation animals, were also getting significant amounts of titanium through their diet. So we pivoted, and the study now is kind of differential of if only ingested, where does it go? And if inhaled and ingested, these are the different tissues that it may be going to. So, but your point about control is great. Well taken.
Yes,
Thank you.
Thank you very much.
Thank you very much. Thank you all for your time.
And MOT number.
Bye. Thank you, everybody on Zoom.
Yeah.